In this video, we'll make a brief presentation of the topic respiratory epithelium and its importance in the system. The respiratory system is constantly exposed, so it must maintain the ability to eliminate pathogens, allergens, and inhaled residues in order to preserve homeostasis. In addition, it is formed by the pseudostratified ciliated columnar epithelium.
This type of epithelium acts as a barrier against pathogens and foreign particles, and prevent infection and injuries throughout the mucociliary elevator. The respiratory epithelium acts as a barrier to protect and clear the airways and lungs from pathogens and irritants.
In the respiratory system there are two portions, the conductive and the respiratory. Conductum. The nasal cavity, trach, bronchi, and bronchioles have a luminal living in the epithelium called cavity cells that secrete mucus to secrete and defense against pathogens. Cells moving in the mucus to trap pathogens. Respiratory portion. They are alloyed with the simple squamous epithelium that facilitates the diffusion of the oxygen and carbon dioxide. The alveolar walls are living by type. The alveolar walls are living at type 2 pneumescence, type 1 pneumescence, and cells that form variform gas exchange and fluid transport. Type 2 pneumescence replacing damaged or injured type 1 pneumescence. Function of the portion. Humidification and warming of the air is achieved by mucus and serous secretion as well as the capillary network in the alveoli that condition and warm the air. Filtration is carried out by trapping mucus secretion and thanks to the ciliary movement that moves the mucus towards the throat for its expulsion or swallowing. Goblin cells secrete mucin glycoproteins, providing moisture to the epithelium and capturing particles and pathogens. Oxidant defense and injury repair. The cells of the respiratory epithelium are constantly combat against inhaled particles and pathogens regenerating after injury. Gas exchange. In the alveoli, the exchange of oxygen and carbon dioxide between air and blood takes place.
Light and electron microscopy is used to visualize the structure of this tissue. With light microscopy with hematoxylin and eosine staining reveals the structure of pseudostratified epithelium in the airways, with goblet cells, basal cells, and ciliated cells on the surface. In the trachea, a light pink epithelium with a thick basement membrane and cartilage rings is prominent. The transition to bronchi is characterized by plaques, and there is a smooth muscle layer between the lamina propria and the submucosa. The bronchioles lack cartilage and glands, and the alveolar sacs, a group of alveoli, are composed of smooth muscle, elastic fibers, and collagen. With electron microscopy, we can visualize the type of cells and ultrastructural features. In the trachea, basal, globid, and ciliated cells can be distinguished. At the alveolar level, the thin air-blood barrier formed by type 1 pneumocytes, capillary endothelium, and fused basal lamina is observed, and type 2 pneumocytes, which are thicker and more robust than type 1, are characterized by the lamellar bodies, rough endoplasmatic reticulum, and Golgi.
Asthma. It is an inflammatory disease that causes remodeling of the airways, generating the hyperactive response to environment stimuli and excessive production of mucus. Inflammation and edema cause bronchoconstriction, obstructing airflow to the lungs, causing wheezing and difficulty breathing. This process involves the interaction between the mucosal epithelium, mast cells, smooth muscle, and the parasympathetic nervous system. Cystic fibrosis. It is a general disease that mainly affects the lungs, causing a chronic and progressive lung disease. It requires early diagnosis and mutation-specific treatment to maintain quality of life. Treatment goals include improving mucociliary clearance, reducing bacterial infections, and improving quality of life.
Another disease would be primary ciliary dyskinesia, known as PCD, which affects the ability of the cilia to move mucus and inhale particles, causing recurrent sinus, ear, and lung infections. Diagnosis involves ultrastructural analysis of the cilia and molecular genetic testing to identify mutations in the 33 genes associated with PCD.